you all for joining us today. This is our first webinar of what we're calling our Grant Writers Toolkit seminar series. And these sessions are put together by the Early Career Professionals Working Group in the RCMB Assembly. And so I'm Jennifer Larson-Casey, and I'm from UAB, and I co-chair this Grant Writers series. And so we are putting on a total of 4 webinar sessions, and they will occur every month leading up to ATS. And so our second session will occur in February, and it will focus on identifying and preparing for NIH grants. The third session will be in March, and it'll look at other like non-NIH grant mechanisms, so DoD, VA, foundation grants. And then our fourth and final session in April will we'll have a mock study section and to kind of take you through the NIH process that occurs when after your grants are submitted. So be on the lookout for those ATS advertisement emails to register for the upcoming events. And so today, this is our first seminar. I was going to share my screen. Oh, shoot. Let's see here. We did this all before. I should have just kept it. Okay, so this is our first, this first session will be on how to build an impactful specific aims page. And we are joined by 2 of the 3 faculty members. And so we want to thank them. We're hoping Scott will join us as we go along. But first, we have Dr. Bethany Moore, who is a professor in the Division of Pulmonary and the chair of the microbiology and immunology department at Michigan. And her research focuses on the immune responses that occur during the pathogenesis of pulmonary fibrosis and post-stem cell transplantation. Hopefully, Dr. Budinger will be able to join us, and he is a professor and chief in the pulmonary division at Northwestern, and he is interested in alveolar macrophages and epithelial cells and how they contribute to the development of acute lung injury and fibrosis. And then finally, Dr. Andrew Wilson, who is a professor in medicine within the pulmonary division at Boston University, will kind of wrap up the impact section of the specific aims page. And his research focuses on genetic mechanisms within COPD. And he is also the scientific director of the Alpha One Foundation research program. So we're very thankful for all of these Dr. Moore, Dr. Budinger, and Dr. Wilson for helping us put together this specific aims page. And so I'm joined by 2 other members of the ECP working group, Chao He, who is at Baylor, and Luis Rodriguez at the University of Pennsylvania, who will be moderating this with me. And then we'd also like to thank Tiana Lino, who is our manager and who really runs the Early Career Professionals Working Group and makes all these webinars happen. And so our agenda today is a didactic session, which will break up the specific aims page into 3 sections. And Dr. Moore will lead with the opening paragraph and hypothesis. Dr. Budinger will take over the aims section, and Dr. Wilson will do the impact session. And we'll have a brief Q&A session, and then we will break into small groups where we'll look at a specific aims page, and that'll be moderated by our faculty members. And then we'll reconvene and kind of discuss what we found in the specific aims page, how it could be better, and then compare it to the original specific aims page to kind of show you how we've kind of altered this specific aims page and ways we could potentially, ways that are potential flaws in a specific aims page. And then we'll wrap up with any last minute Q&As. So with that, Dr. Moore, would you like to take it away?
Yeah, it's my pleasure. Well, welcome everybody. So I guess today we're really focusing on the specific aims page. And so I thought I would just start off and say, why is this page so important for your grant application? And so for those of you who may be new to this process, you may not realize, but when you send your grant to the NIH, it's going to be assigned to 3 people who are going to review it. But there may be 25 or even 30 members who are on the study section panel that are going to listen to that discussion. And so as sad as it is after you spend all that time preparing this application, it really gets discussed in something like 10 to 15 minutes at this section, and really only 3 people are going to talk much about it. And so the primary reviewer is going to briefly outline your grant. That means they're going to talk about you, the institution, the aims, the impact, the significance, and briefly what your methodology is. And that's going to hopefully happen in about 5 minutes. The secondary and tertiary reviewer are asked to only really highlight what the strengths and weaknesses are. They're not asked to go back and rediscuss or re-outline kind of what the aims or the methodology were. But then what's interesting is that at the end of that discussion, at that 15 minutes, all 3 of those reviewers are going to suggest a score, but everyone on the panel actually scores the grant. And so they're going to decide whether they want to agree with reviewer 1, 2, or 3, or whose discussion, carried the most weight for them. And so really what that means is that everybody else on the panel that's not one of the 3 people who actually read the grant, they're going to be sort of listening with one ear, and they're going to be looking at your specific aims page during that discussion for the rest of the grant. And so the clearer that your specific aims page can communicate what you're doing and what your plan is, the easier it is for the other people on the grant panel who did not read it carefully to be able to, get excited about it, see what it's going to be. And so it's really important for it to be super, super clear. So that's really the best way for everybody else to kind of get the rationale, the methods, the aims in less than 15 minutes. And they're going to be reading that super quickly while they're listening to somebody else talk. And so the clearer and easier it is, the better. Next slide. So if we talk about what's needed for your grant's aim page, really what you want to do is you want to make sure you state the global issue that your study is going to address. And then, so that's usually the disease focus in many cases, especially for ATS related grants. You're going to have enough background just to give context for what part of the puzzle you're going to try to address in your aims page. It's important to identify what the gap in knowledge is because, of course, study sections are asked to really fund innovative work that's going to do something meaningful. And so if it's just rehashing a topic that we already feel like we know about or that's been studied for a very long time, you're really going to have to work to highlight why you still need to do this work. You're asking for more money to keep studying that same problem. You're going to have room to state just a few of the key findings. You can't do a thorough literature review on the aims page, but you can just put a few key findings, usually put a few pieces of your key preliminary data, maybe even in bullet points or a numbered list. You're going to clearly state your hypothesis, and you'll see when we talk about the aims page, it's important for that to be super easy for the reviewers to find. So bolding it or underlining it is a good plan. You're going to have 2 or maybe 3 very actionable aims. And you're going to remember what the timeline of the grant is. So if you're writing an R21, you've only got 2 years to do those aims. If you're writing an R01, you might have 5 years to do those aims. And so making sure that you propose work that will be reasonable within the time frame of the grant is another thing that's important. I really love a summary figure on a grant. It's optional. I think it's great when it works, well, if it can really help highlight what you're going to do, but it will show you some examples. If it's not clear, it can be the worst thing ever. And what sometimes in the aims pages, so there's so much information you're trying to put on an aims page, sometimes you have to put that summary figure actually into the body of the grant and then just refer to it on the aims page. I've seen that work as well too. And then at the very end of the specific aims page, I like a summary statement that really says, what am I going to learn if we finish this? And what's the impact of this grant? But as you can imagine, even with 11-point font and 0.5-inch margins, this is all a lot of information to still try to fit on a page. So next slide. So here's just a little diagram of sort of what the structure of an aims page will look like. And I'll give you some other references. And I believe y'all are going to get these slides afterwards, so don't feel like you're having to try to scribble things down now. So again, the opening sentence, you're really wanting to frame your topic in terms of its importance to the NIH or the foundation that you're writing for. Then you're going to next, briefly summarize sort of the current knowledge in 2 to 3 sentences to sort of lay the foundation. It's then good to identify what your critical knowledge gap is and, maybe underlining that or bolding it. I've mentioned that sometimes if it's a training grant, you might also highlight what training will be gained in that case. And then you can have room to outline a few key pieces of preliminary or published data that's going to form the basis of what is a novel hypothesis. And then, like I said, that can be bullet points or a numbered list. And then provide the rationale for your hypothesis. And some people like to put an overall rationale there. Your central hypothesis, I think, needs to be something really clearly bolded. And then say that you're going to test that hypothesis in these, 1, 2, or possibly 3 aims. And then, as I said, under those aims, you're going to give a title. And I think one thing that's really important is the title of your aims, that they need to be clear about what you're going to do and actionable items. And then, like I say, a picture or diagram can really help. And then that final sentence, I think, really highlighting how if you explain that after you complete these, what's going to be the benefit of having done all this work? Why should the NIH give you taxpayer dollars to be able to advance this work? All right, next slide. All right, so here's just to give you some example of what I mean when I say that your opening sentence, this is really what the global issue is, or some people like to call, you'll see on the next slides, they call it the hook. And so you're really trying to frame the significance of your work. So these are just examples. I'm not saying these are the very best examples. They were just examples from grants that we use in a grant writing course that we do here. Stem cell transplant can treat a variety of genetic defects and malignancies, but it also has a lot of pulmonary complications. So that's the problem. This grant's going to be about the pulmonary complications and how can we address those. IPF is a devastating and fatal scarring disease. We don't have effective treatments for it. Are there unchecked fibroblast pathways that we could target for therapeutic benefit? So, you can get the idea. So the idea here in this opening sentence is really to kind of frame the biology question or the disease question that you're going to answer and set up what it is that's important, why it's important for you to do this work. All right, next slide. I don't think I need to read all of this to you. And then in the next section of it, you're really providing some background or the context or the key findings. And so some examples of how you transition from that background into the preliminary data or that gap in knowledge, which is sometimes called, this is where you can say, our murine studies have shown that, pharmacologic or genetic inhibition of COX-2 can restore innate immunity in vivo and alveolar macrophage function in vitro. We're going to present, preliminary data that and then we're going to give them a few pieces of our preliminary data and then say that the gap in knowledge there is that we don't know how PGE2 signals to impair that innate immune function or whether inhibition of that pathway might improve function. So this was obviously a grant where we were studying prostaglandin signaling as an inhibition of host defense. And the next one, this was from a grant where they were really trying to do serum biomarkers and looking for a biomarker profile. And so, they kind of transitioned from the fact that IPF is a disease that's hard to treat, hard to prognose, hard to diagnose. It's unlikely that any single biomarker will be powerful enough to provide accurate or prognostic information. And so the gap in knowledge that they're trying to address in this grant was whether assessing multiple extracellular markers, simultaneously could really give them this fingerprint or a signature that would be useful. And so that was how they were going to lay out the aims of that grant. So just trying to give you an example of sort of a basic science context and a more clinical context. Next slide. So then when you get to that next thing, now it's time to state your hypothesis. And what I like to say is, you really don't want to be ambivalent about it. And one of my key pet peeves, I guess I would say, when I'm a grant reviewer is if I read the hypothesis and it does not really predict or tell me what they think is going to happen. And so that's what happens. So here's just a few examples. And so this was a grant on periostin that I wrote back in the day. But so what you want to say is that matricellular, I'm hypothesizing that periostin actually promotes fibrosis. I'm not going to say that it alters fibrosis because a word like alters doesn't tell you whether that's, is going in the positive direction or a protective direction. And so the same thing, like if you get down to, on the next one, we hypothesize that an RNA virus upregulation of TLR9 contributes to overexuberant cytokine response. I don't want to say that TLR9 alters the cytokine response because then you're leaving the reviewer to try to figure out whether TLR9 is making it better or making it worse. If we're talking about epithelial cell macrophage crosstalk there, I want to be clear that I think it impairs bacterial phagocytosis and killing, not use a word like change. So that's just to show you that the language that you use in the hypothesis, I think it's good to be very clear because remember, you're testing your hypothesis. It's okay if what you find is that the hypothesis was wrong. You're still just testing it. So be clear in what you anticipate and then test to see if that's correct or not. All right, next slide. And then this is where, like I say, I always tell people, one of my suggestions when you're writing a grant, you should try to diagram it, because if you can diagram it, even if that diagram never makes it into the aims page or the grant, you really gives you clarity of thought about the grant. Because if you can really lay out that schematic of what the grant is doing and what it's testing, what the signaling pathways are that you're interrogating, then it means that you've thought carefully about it. And it's usually a good way to really catch if you've got a hole in the grant, if you will. So like I always say, it's a picture's worth 1,000 words. And like I say, if you can't fit it onto the specific games page, sometimes putting it in the first body of the grant, that can work really, really well. And again, I think the reason why the picture is particularly helpful is because it's a visual that if it's easy to interpret, it gives those other reviewers around the table something to focus on while someone is discussing the grant, and they can easily see, oh yeah, aim 1 is going to be in this part of the pathway, aim 2 is going to be this, aim 3 is going to be this. I think particularly if you're studying a signaling pathway, Wnt signaling or, a metabolism pathway or something like that, those are not going to be familiar to everybody around the room. And so if you're really studying something that's a detailed signaling pathway or metabolic pathway or something like that, I think it's good to remind people what, don't think we're going to remember our basic biochemistry or, I'm studying the tryptophan pathway right now. I guarantee you, I still need to look at the diagram all the time to remember which things come in which order and which enzymes catalyze which conversions and things like that. So don't make your reviewers hunt for that. Give it to them. All right, next slide. So here's just an example, which is funny to go back and look at it. So this was an example of that bone marrow transplant grant that we wrote where we were studying PG2. In this grant, what we thought was happening is that the epithelial cells were getting injured in the transplant. They were releasing TGF-beta that was causing all of these epigenetic upregulations of microRNAs and altering DNA methyltransferases, getting COX-2, making more PG2, that was binding back to the EP2 receptors, and then that was inhibiting phagocytosis and killing. So a pretty complicated pathway that I think was much simpler for people to read this grant because we diagrammed it out. If I look at that hypothesis right now, I think in retrospect, that hypothesis was way too complicated and long, probably. So I would work on that. But I think the picture probably helped here for people to be able to see that this is the pathway that we think is true. And then you can see, aim 1 was going to be about inhibiting the COX pathway. You can see aim 2 was down there looking at the role of TGF-beta in driving the microRNA phenotype, and then aim 3 was going to be about the autophagy and killing defects. And we had some therapeutic in aim 1 on the PG2 side on the EP2 receptor as well. So I think in this case that this figure probably helped the reviewers understand sort of what I was proposing to do, because that's a lot of complicated, biochemistry for somebody that doesn't think about prostaglandin signaling. Next slide. And then, we still have that pathway has served us well. We've studied it in diabetic wound healing as well as in bone marrow transplant. So here's an example of us kind of diagramming that out again. And the reason I wanted to show this is that our figures got a little bit better, when we learned how to use BioRender. And B, what we did here is we tried to put some sort of colored boxes that kind of highlighted, which part of the pathway was aim 1, which part of the pathway was aim 2, which part of the pathway was aim 3 for this. And in retrospect, this hypothesis was probably not quite detailed enough, but I think, again, the picture helped because you could still follow the plan of what we were planning to do. But like this grant was all about that MLL1 enzyme regulating that, histone modification. And I don't even mention MLL1 in the hypothesis going back. So even I learn as I teach these grant review panels to go back and even be more critical of what I wrote in the past. Next slide. And then this is, again, an example. This is not my grant, but one that was from Eric White's lab at one point in time, where this was really a grant that was more meant to be like sort of a process of how they were going to look for a biomarker. And so what he did in this, and I think it worked really well, was rather than testing a particular hypothesis, he was just highlighting like what the steps were that they were going to go through, what this grant would entail, that they were going to draw blood and have tissue. They were going to do these evaluation of these biomarkers, and then how they were going to look for it. And so I think that also made it very, very clear sort of what they were planning to do over the scope of that grant. So this is just sort of another example of how you can use a figure to sort of lay out what the grant hopes to do. Next slide. All right, and then I just wanted, so basically everything I just told you, this is just looking at it a different way. So this is from this grant application anatomy of a specific games page. So that is an online tool that you can go to. And I think they do a really nice job. They give you an example of a grant here. And what they do is they color code the pieces of the first paragraph and what they sort of relate to. And so there's the hook about, cancers and vaccines and malignancies, viruses and cancers worldwide. The known information kind of talks a little bit about what they know about HTLV. In that case, they highlight what the gap in knowledge is in the yellow paragraph, and then the critical need there is sort of what they still need to do. So I think if you can, think about laying out your first paragraph sort of with this hook, what's known, what's the gap, what do we need to do, why do I need to do this grant? That'll help you frame what that first paragraph really should look like. Next page, next slide. And then in the second paragraph, because when you're transitioning from sort of that background introductory piece into sort of what you're going to do, then here in this example, which I think is still similar to what I diagrammed before, they're laying out in the red there what the long-term goal is, the proposal objective, and then the rationale. That's a very important thing. Why do we need to, what makes us think that this pathway we're studying or this thing we're going to investigate is worthwhile? And then the hypothesis is there. I would have said I would have liked that hypothesis bolded or underlined so it's even easier to see. And then in this case, they're kind of highlighting what the payoff would be or the impact of the work. Next slide. All right, I'm going to hand it off to Dr. Budinger.
Great. Thanks for, sorry I was late. I'm Scott Budinger. I'm the Chief of Pulmonary Critical Care at Northwestern. I think I'm on this panel because I've reviewed lots of grants like Beth has. And happy to talk a little bit about the specific aims, which is really the meat of the grant. What I would, I think Beth already mentioned this in her remarks, but I think it's important to realize that in this room full of people that are reviewing your grant, they are going to be looking at that first page, which will be open on their computer as somebody is discussing and introducing this grant. And they will read your specific aims, if nothing else, right? And so you want these to be, as much as you can, you want them to be causal, right? And, so if you are doing a mechanistic grant, you would like to highlight how your experiments are going to show gain or loss of function, preferably in a genetic system. And sometimes you can't do that. Right. And if you can't, and this for the example would be if you're doing an observational study in a clinical cohort, right, that can't be causal, it's going to be an association. And there you should acknowledge that you're going to find an association. So be clear about what you are actually going to be able to test and conclude. You also want this to be as granular as possible. And we'll give some examples here, which I think will help to do that. But Beth mentioned her example of osteopontin. You want to actually say, I think osteopontin is working on in this cell to, if you can, to affect this receptor downstream to modify fibroblast activation in that example. So very, very granular if you can, because again, that gives the reviewers and the people that are going to be scoring your grant without ever having reading it, some information about exactly what kinds of experiments you're going to be doing. Cell type specific, you'd like to have as much specificity as you can around the cells that you're specifically targeting. And if you can in the grant, you want to link this to the clinically relevant question. So in the example of that Beth gave to pulmonary fibrosis. So in mouse models of pulmonary fibrosis or in patients with pulmonary fibrosis, for example, if you're doing this. Again, sort of tying this back to the significance piece of the evaluation. Next slide. So we're going to just go through some examples because I think this is the easiest way to do this. And we're going to talk about how we can start with some things here and then maybe talk about how we might clean them up. So these are actually made up. So these are not real. These are sort of taken from some published work. And I'm going to give you, I just want you to read these aims and I want you to tell me what you think about them. I don't think we can actually do a poll here, but just think of what grade you would give this aim. So we've got, to characterize the role of macrophages in the development of pulmonary fibrosis. To determine how macrophages are related to epithelial injury in lung fibrosis, and to test the role of macrophages in patients with pulmonary fibrosis. So for me, these would be atrocious aims, right? These are terrible. They say things like characterize, which is a dirty word, right, in the aims. They are not granular, they're not causal, and they're not specific. So let's go to how we might think about revising these and just give you an example of what I mean by this. Next slide. So here we're actually getting to something that's a little bit more causal and granular. So to determine whether signaling via the CSF1 receptor is necessary to maintain monocyte-derived alveolar macrophages in fibrotic lung lesions in murine models of pulmonary fibrosis. So you can see how we've gone to sort of the general, vague, and not very helpful to actually something where you can get an idea about what experiments are going to be done in these by this investigator in these specific aims. And then this comes from Beth's work to determine whether alveolar epithelial cell production of CCL2 is necessary for the recruitment of monocyte-derived alveolar macrophages in murine models of lung fibrosis. And then to determine whether expansion of CSF1 receptor expressing monocyte-derived alveolar macrophages is associated with fibrosis progression in patients with scleroderma-induced lung fibrosis. So again, quite specific and actually very clear to somebody that's reading these, how this works. And if you go to the next slide, and this is something that you can choose or not to do. Very often what people will do is to actually have a piece of text, and I'm not going to read this all, you'll get these slides, have a piece of text under this that actually shows that you really are going to be doing this in a causal system. So, here in the first name, I put, we will inducibly delete the CSF1 receptor in labeled cells and examine fibrosis severity in the bleomycin and asbestos models of lung fibrosis. And unlike I did on these slides, you should actually check to make sure you spell everything right. But these are examples of how you might go to to really give some description to those people. And again, the audience that you're going for here is not necessarily it is your primary reviewers, but it's also those people in the audience. They're going to look at this paragraph in your grant, and that may be all they see in addition to a couple of the figures that you have in the rest of the grant. Next example here. Next slide. So again, here we've gone to just to sort of put this home. Here we've got some examples, again, to determine whether mitochondrial ROS are produced by macrophages in response to ambient particulate matter air pollution, to determine whether inflammation in alveolar macrophages is responsible for cardiovascular events, and to determine whether beta agonists worsen inflammation in alveolar macrophages to drive cardiovascular events. These are, again, bad aims. Next slide. So we can actually so this is a way, again, just the example here, to determine whether ambient particulate matter air pollution activates calcium release activated calcium channels in alveolar macrophages to drive the systemic release of IL-6. They're both describing the same experiments, but here this is a much more granular description that actually gets you to, you can start to imagine in your head the experiments that are going to be proposed to actually test these hypotheses. And similarly down the line here, I won't read these all. Next slide. And again, we can actually then put in very specific information about what we're precisely going to do within that specific aims page. And again, give a nice summary for the reviewers of the grant. And again, you may actually find some investigators that you are working with if you're working on a mentored grant, or you yourself may choose your own style here. Some people will actually just put the aims without putting the subtext. Others will use the subtext. I find it helpful, but it's going to be a style issue. And then finally, the next slide. And this is an example of a 2-aim grant where you would be looking at a clinical type of proposal where you might not have actually the ability to do a causal experiment or to get to that granular mechanistic level, but you still can be a lot more, provide a lot more information often in the aims than is often provided. So here we have to determine whether a conservative or liberal fluid strategy is superior in patients with sepsis, and to determine whether biomarkers of sepsis severity are reduced in patients receiving a liberal versus conservative fluid strategy. And if you go to the next slide, we can really clean these aims up by actually being, again, more granular, more specific. So to determine whether fluid resuscitation with lactated Ringer solution using a conservative and then being precise about what that is versus or liberal strategy is associated with improved clinical outcomes in patients with sepsis or septic shock, right? And you give a definition of what you mean by sepsis and septic shock. You can actually put in this subtext how many patients you're looking at. You get a nice sense of what that is. And then to determine whether changes in lactic acid levels, PF ratio, and BNP can be used as biomarkers to guide fluid resuscitation. Again, a much more granular aim. So I think this is something that you should, working on your specific aims and your specific aims page should be something that you do early and often. If you're thinking about submitting a grant in the summer, this is the time to actually have a specific aims page and be circulating it to all of your friends and actually getting a lot of feedback on that, because this will force you to organize sort of where you're going to be going with the rest of the application. And I'll stop there. I think we're going to have time for questions at the end.
Okay, great. Okay, so at this point, thank you all. Thank you, Beth, and thank you, Scott. It's my pleasure now to speak about the very end of the aims page. So at this point you've You've introduced the topic, you have identified the knowledge gap, and you have stated your clear and testable hypothesis along with your causal, granular, cell type specific aims that are linked to a clinically relevant question. And you've done a really nice job with all of that. And now you're at the very end of the page, and you have the opportunity to sort of leave the reader and the reviewers with one last thought. And so you have it's an important part of the page. I think people always read the end of the page. So if you have real estate left at the end, it's really nice to have a paragraph that's at least 1 or 2 sentences long that follows the last aim. And the idea here is to frame your project again in the mind of the reviewers and remind them why this work that you're proposing is so important. And so I think that the main thing to think about when you're doing this is to think about what the big picture goal is, or the significant knowledge gap that you're addressing, and stated in broad terms, not in very detailed terms. So you've gotten really detailed with your aims and your experiments that you've outlined, and now is your chance to sort of frame it again in broader terms about the impact. And in so doing, it's nice to outline kind of like broadly how the work will be impactful for the field or the community. Next slide, please. So this is just an example of how that might look. This particular example is a little longer than I would typically do for this kind of a paragraph, but they say in the first sentence, the proposed studies will establish a new mouse model that will overcome current limitations. So highlighting the innovation and provide greater insight into the mechanism of HTLV1 tax tumorigenesis. Knowledge is currently lacking, and it promises to yield novel insights into viral and cellular biology. So they tell what the expected outcomes are going to be. And then the new and improved mouse model for tax tumorigenesis will provide a valuable resource for the wider scientific community to pursue a multitude of studies that have not previously been possible due to limitations of existing mouse models of tax. So that's the impact of the proposal. Framing it into how it's going to be useful for the entire field. Next slide, please. I just wanted to, this is not the way that we were sort of primarily recommending, but I have read a lot of grants and seen, there's more than one way to write a grant. So this is sort of another way to kind of accomplish a similar thing. And if you have something about your team or the science that you're doing that's just really specific and unique to your center that you want to highlight. And maybe, and the example I give is one from a grant that I wrote for the American Lung Association during COVID. So I didn't have to convince anybody at the time that COVID was important, because everybody was thinking about nothing else. This is probably in about June of 2020. What I wrote instead was our combined expertise in iPSC-directed differentiation protocols, lung disease modeling of environmental exposures, and positive sense RNA virology, together with unmatched resources described above, uniquely positions us to address a critical knowledge gap of high interest to the ALA and the lung community. So the concept here is that we just have these unique resources. We are better positioned than anybody else to address this really important question. So that's an alternative that you could do to sort of get to the same place, basically leaving the audience with a statement about why this is so important and how it's going to be important to the field. And I think that's all I have on this topic.
Yeah, go ahead. Do you want me to take this one? I can just say this was just to reminder that as the reviewers are reviewing your aims page and listening to it, these are some of the questions that they're going to be asking. Are they going to be able to get the work done in the project period that you've proposed? Is the work too overambitious? This is a very easy thing to criticize, especially in early career grants. And if you have to breed a whole bunch of complex mice, you don't know for sure what phenotype they're going to have, things like that. Those are always things that people are easy to pick on. Did the PI describe the potential pitfalls and the possible alternatives? Will the experiments generate meaningful data? Will the resulting data prove your hypothesis? And these are more about the research design than they are about the aims page. But as you're thinking about your aims page and what you want to convey, these are the things that the reviewers are going to be sort of reviewing at the very end. Whether others are doing the work or it's already been completed, and whether you've, addressed a rigorous sample size and all. So again, much more for the research design aspect, but just something to keep in mind. Sorry, now I think that's the last slide.
Yeah, so we're happy to take any questions. I think there were a couple in the chat. Chao, do you want to ask that question?
If you have any question, you can type in the chat, or we'll have a Q&A session at the very end. We'll take some questions. Now, there is one in the chat that from Emily Huffman. So she asked a question about maybe it's a new NIH guideline or recommendation. I think maybe I'll share this. So I share something else. So she asked this NIH page is about regarding figures, and highlighting this part is that figures should not be included in the specific aim attachment. Figures can interfere with the report system. I don't know any of the.
Yeah, well, I can take that. I can take that. So first of all, I just want to thank Emily for highlighting that to me. I had not seen that. I just finished reviewing grants, and a lot of them did have figures in the specific aims page. And then Emily and I were having a discussion in the private chat, and she said that she was able to submit a grant with it on the aims page, but she got a warning from NIH saying that it could interfere with the post-award process. So I didn't know that. So I guess what I would say is I will go back and I will modify my advice in the future and say that putting that figure should probably happen into the, at the beginning of the research design instead of on the aims page. If you don't know what the reporter is, that's just a place where you can go and search to see, like, when you're looking to see, when I was looking to see where I wanted to go postdoc, I went, to such a thing, and to see if the people I was interested in had grant funding and things like that. So it's just a place where you can search and see, what kind of grants have already been funded in a particular area. You can search by name of PI and all. The aims page doesn't usually show up on that. To my knowledge, it was always just the abstract, which has no, you can't put the figures in there. So I am surprised to see that guidance, but we may get a lot of confusing guidance in the next little bit. But if they're saying that, then I would say put your figure into the body of the grant. So
stay tuned. I just submitted some grants with figures on the first page, and they went in.
Okay, I think there's another question. Go ahead.
There's another question from Lysandra Ellis. For basic science grant, do you have any specific recommendation in terms of clinical relevant questions? I think this probably also kind of the hook sentence that you write at the very beginning, Beth mentioned, kind of putting a disease context, but if someone is doing really basic science study, may not have a very broad application, but not specific to one disease condition. If it is you want to kind of address your question specifically, you can unmute yourself.
Yeah, I'm sorry. I'll just say that I didn't mean to suggest that a disease relevant grant is the only thing that does well at NIH. That's not true. Basic science definitely can do well too. And I would say that what's important there is you still want to frame the importance of the biological question that you're asking. What's important about the why is it important for the question? And usually you can frame that in if you want to unmute and tell us what an example is, we can try to give you some feedback. But usually you can still frame the importance of your work in understanding a basic biological process that's important for the physiology or the turnover or the metabolism or the proliferation of cells, and that could have implications for cancer vaccine physiology, something like that. You can usually come up with some way to frame it in the larger context. But a biological question that needs to be addressed is just as important, I think, as a disease relevant question.
But and this may be like everything might be dated advice, right? But at least now, for now, we NIH has I don't remember what it is, institutes, right? So it's 20 odd institutes. Those institutes have different diseases that are actually in those institutes, and they have different pay lines. And so for a very pragmatic reason, if you are studying a basic biologic question that has a particular relevance to a given disease, you want to highlight how important it is to that disease, and that's important. So there are grants, as Beth said, that it can be fundamental biology. General medicine actually funds those types of grants. Those study sections have very different reviewers, different pay lines, different budgets. And so you really need to think about, if you are writing to a specific institute, they are mandated to actually fund a disease that's relevant to that organ system or that disease, and you do have to tie your biology to it, and that will be score driving criteria for the reviewers who will score you on significance, right? And if you don't actually prove to them that that's significant for the disease you've linked it to, you'll have a problem. So it's not a trivial issue. It's an important issue, Lysandra. It's something that you do have to address. And this is somewhere where, having disease experts or clinical colleagues or collaborators can really be helpful to you if you're not, if you don't feel comfortable making those kinds of statements. I don't think you have to register for a different website. If it gets funded, it will automatically go into a different website that Beth is talking about, which is NIH eReporter, where you'll be able to look at actually funded grants by the NIH, and it will include your project summary, your abstract, essentially, so that others can look there. And it's oftentimes useful to browse that in your field, right, and look at what kind of grants the NIH has been funding or the study sections have been interested in funding in your biologic or disease area.
Okay, well, thanks for all answering those questions, and thanks so very much for the presentation. I think we're going to switch over to a bit of a more practical application. So there's a large number of people in the room, and we have breakout rooms planned out. So everyone here is going to be transitioned over to one of 3 breakout rooms where you will work individually with our 3 moderators. And what we'll do is we'll look at a specific AIMS page that is a variant of a successful specific AIMS page, and we'll try to identify this AIMS page has been changed a little bit to be less effective, and we'll try to look at perhaps what is less effective and maybe have a conversation about what can be changed and how we can make it more effective. Yeah, so I think at this point we can go ahead and transition over if Tiana can take care of that for us. All right.
So I can share my screen again, and we can kind of look at the unedited, the original funded specific AIMS page, and then the one that we all just looked at. And so we can kind of get just a brief overview of kind of the difference in these. And it's kind of hard to share, but I guess if someone would like to go through kind of or talk about the differences that they just see visually, kind of, the overview of what you see that's different between the original that was funded on the left here and then the edited one that we just discussed on the right.
Text is all justified, and there's bolded sentences that immediately catch the attention, and the spacing is better. Like there's no space after the aim and the description of the aim.
I was much harsher than the review panel on this. I didn't like the aims here because I think they could have been more granular. Again, they're very broad.
I think some of the other things that we noted in ours, in this grant, you could make, again, this was a funded grant, so it did well. But I mean, in this grant, it was pointed out that, AIM 1 seemed to be critical, and, AIM 2 and 3 might or might not be necessary the way it was laid out. And that's always, the dependency of the aims was something that got highlighted in our group. And then, I know it's hard to read here, but in the one that was kind of modified, I think it was highlighted in our group that they really wasn't placed in the context of a biological question or a disease question, and that sort of, introducing the concept and why it was important and why this needed to be done just wasn't clear, and that the whole rationale paragraph was a lot of sort of repetitive stuff that still didn't, a lot of real estate to not really get to the clear point about this being a grant about lung regeneration potentially, and that being what was important about it. Things like that.
I have a question. Is a title something that you want to include in your specific AIMS page?
I've seen it both ways. I'd say it's less common than not, just because usually people don't have room, but I have seen titles on a specific Ames page, the title of the grant sometimes listed.
Oh, yeah. I think uncommon, generally. Less common than common.
I would say less common, yes. I think we can all just point out that in the grant that was funded, you can see that the hypothesis is bolded, which does really help. It's really on the one that we mucked up a little bit. That figure is not a helpful figure. And the hypothesis is very hard to find. So if you're the reviewer who's scanning it, at a minimum, you want to highlight the hypothesis word, if not the whole sentence.
Similarly, I had a question like, should we be stating the overarching goal? This is a mechanistical study. I'm not very familiar, but should we be including something like an overarching goal of this, whole proposal?
I can address that. Yeah. I mean, I think it's a great idea to do that. And I personally like to make it really clear within the first, as early as possible in the first paragraph, what is this grant about? So that the reviewer who's reading it, maybe and only going to read it once, very quickly understands what the grant is about without having to read through the entire first paragraph or second paragraph to get there. So yeah, I do think it's a good idea to try to have your overarching, whatever the overarching theme or problem is early on.
Yeah, I think if you've ever seen that grant writers workbook, which is a common workbook that's given to people, they recommend highlighting, the overarching goal of this application and bolding that, and then the specific goal of this application. So sometimes that's like framing, like eventually we want to learn how to regenerate lung. What we're going to do in this grant is figure out what regulates AT2 to AT1 differentiation after injury, something like that, so that you really highlight what the specific goal of this grant is versus your overarching theme is a way that they talk about structuring the Ames page.
I think a lot of times that can come after you've identified the knowledge gap also, or the problem, right? It's a natural time to say how you're going to be, or what you're going to be addressing.
I have a question. You briefly mentioned something about the aims being dependent from each other or independent from each other. Can you elaborate a little bit more on that?
Yeah, I can say, a lot of times people will tell you that if you're reading a grant and if aim one fails, if your aims are dependent on each other, what that means is that if in aim one you get a different answer, then is there any value to still doing aims 2 and 3? So I do think you want your aims to be related. They're usually still around a central theme or something, but what you really want to do is make sure that aims 2 and 3 are structured such that there's still useful information to be gained there, even if aim 1 is negative, that there's still something else that's important to study there. So that can be a really hard thing for novice grant writers to really figure out the dependency of the aims. But I guess what you could think about is as you're reading your grants and thinking about it, ask yourself, if I get completely the opposite answer in aim 1, if my hypothesis is completely wrong, would I still do aim 2 and would I still do aim 3? And if not, try to restructure aim 2 and 3 to be something that even if aim 1 is wrong, would I still be able to go this direction? So usually as long as you're focused on, like sometimes I'll see a grant focused on, one aim will be on like epithelial cell. In our grants, a lot of times one aim is an epithelial cell grant, one aim is a macrophage grant. I might be looking at similar questions in both, but they're unique from each other. Even if the answer is different in epithelial cells, maybe the macrophage part will still be true. So I don't know if that helps frame it. I don't know if Andrew or Scott can put that in better words than I can.
No, I think, Beth, you said it exactly right. And this is something actually where we're getting to that causality, that necessity and sufficiency can actually be helpful because those two things are usually not mutually exclusive, right? And so if you have a nice approach that looks at necessity and then looks at sufficiency, right, that's oftentimes a way that you can reorganize things to make aims not dependent instead of putting those in the same aim and then going somewhere else.
Yeah. Sorry, go ahead.
You, Andrew, first, and I'll mention.
I was just saying an additional sort of another way the problem can arise is if aim 1 is going to identify something like a list of genes or something, and then aim 2 is going to look at mechanisms. So, and I have a postdoc who's writing a grant right now in which that's the case. We're going to identify genes in aim 1 and look at the mechanisms through which they affect the clinical problem in aim 2. And so to try to get around this issue, the way that we're having him write aim 2 is that he's also going to pull in genes from the published literature, so that we're, even if, let's just say that we don't get the gene that the genes from AIM-1, we'll still have something to study in AIM-2 because there is data out there that we can pull from. And so that's sort of another technique. You just want to take that idea out of the reviewers' minds as explicitly as possible early on, because otherwise, as soon as they think there's interdependence, if they form that opinion on the Ames page, they might not read all the way to the end to find out that, oh, you actually have a plan for this.
Yeah, so another question I get asked a lot is that, how much, data do you have to support? Is it okay to go ahead and make a suggestion that, you're hypothesizing, this particular directionality of your data? And I say yes. I mean, so if you've got preliminary data that already kind of gives you a pretty good sense that, these two things are important, then they're not going to be as troubled by the dependency of the grant because you've already shown them, data. Like if I'm just trying to think of an example, if you've got preliminary data that there's two different transcription factors or two different cytokines that are important in something you're studying, then I don't think you'll get in too much trouble with, one aim being about one cytokine and the other aim being about the other or something like that. So data can really help because if you've got strong preliminary data that really supports what you're planning to do, you don't seem to get those dependency questions quite as often because you've already sort of provided some evidence that you're on the right track.
Well, if there aren't any more questions, I think this was a really great session. And
Actually, I have one question. Sorry. It's about a technical question. So, how to decide how many specific aims we will put? For example, for these specific aims we discussed, the AIM-2 and AIM-3. The AIM-2 is about what if the gene is knocked out, and AIM-3 is about what if the gene is overexpressed. What's the outcome of these two methods? So I'm not sure if we just put these two aims as AIM-2 and overexpress and the sub-aim and overexpress and knockout as the sub-aims, or just we put them as three independent aims.
I will say it generally takes less room in the grant to make it one aim and describe. I mean, first of all, I just want to say that in mechanistic questions, I always like the idea of a grant where you knock something out and then you try to overexpress it. So the knockout and the add back is always a good format really for mechanistic grants. So I do like that. It usually takes less real estate to have two aims than three aims if really you're doing the exact same questions, but you're just using a knockout mouse versus an overexpressing mouse or something like that, because then you don't have the real estate of having to re-describe all of that methodology again and again and again. So in that case, I would say two aims is fine. I don't have a problem. I mean, you can do it either way, but I think it's easier. Remember, you've got to have room. And the thing that I think early grant writers often struggle with is they don't spend enough time on the anticipated results section, because especially for reviewers that don't know your system well, telling them what you think you're going to get is a super important part. And that always gets the short shift in it. And so I think if I would rather see you put those two aims together and spend a nice paragraph about really, what we'll see when we knock it out and how we're going to expect to see it differently when it's overexpressed, I'd rather see you spend your real estate that way probably. And it also looks less ambitious because over ambition especially for young grant writers is one of the easiest things to criticize. So
I would echo that, Beth. I think that you should, don't feel that you need to have three aims. You can do a two aim grant. If you're writing a K, I really would encourage you to do a two aim grant. It's very difficult to actually pack three aims into a 6-page grant. And so think about ways to do that. The exception would be, if you have a gain and loss of function like Beth suggested, and you maybe have something that's human, that's going to be some sort of human correlative that you can't pack into the other two aims, or it doesn't fit really because it's a little orthogonal to what you have in the aims, then it's okay to have a short aim that might describe how you're going to credential your findings in a human system. But in general, I think, particularly as Beth said, young investigators should not be afraid of doing a two aim grant. It sort of knocks the ambition question out of the park right at the get-go, so.
Thank you.
I guess I'll just fill some quiet space by just saying that in my experience, the best way to learn how to write grants is by writing them and also by reading them. So to the extent that you can read your colleagues, grant submissions or your PI's grant submissions or anybody else's, grant proposals, that's a great way to to sort of see how different people do it and get experience reading games pages and trying to analyze them is really helpful. So if you're on a review panel, that's obviously the best. You get to read tons of grants, but short of that, read your colleagues' grants.
Yeah, read and get your colleagues to read yours. Do not be shy.
I will say just one other thing about the specific games page that didn't come up in conversation. This grant has a little bit of alphabet soup in it. I think we can get very easy for us to slip into using all these abbreviations for things that we know, AEC1, AE2, PRC, things like that. Be careful about doing that again, just because not everybody. So, use the abbreviation that makes sense. But if you can, try to limit it to just the 1 or 2 key abbreviations, because it gets very hard for reviewers to remember what was abbreviated.
Absolutely.
Well, I think that's a great note to end on. And thank you, Dr. Wilson, Dr. Moore, Dr. Budinger for participating in this session and providing your insight and knowledge. And thank you all for participating. This was really a great session of interaction. And so that makes it a lot more fun. So thank you all for attending, and look forward to seeing you guys at ATS, if not sooner, at one of our grant writing sessions leading up to ATS. So thank you all.
Thanks.
Thank you.
Bye now.
Everyone.